All right, hi everybody. Today we're going to talk about the objectives, the learning objectives that are printed by the College Board for your cell test on part 1. So there is a second cell test, and that comes at the end of these, and so you'll skip that. But we're just going to talk about these learning objectives right now. I'll say a couple things about the learning objectives. I know that listening to a video on learning objectives is not at all exciting. However, I will say that I think they're super important. My daughter has taken about 10 AP courses, and she tells me that she now takes notes on her learning objectives so that when she goes to test, she has all her notes right on the learning objectives. So just something to think about. You could open my presentation on one side of the screen and have your own learning objectives on the other side. You could make a copy of it and write all over it, highlight it, do whatever you need to, add pictures to it. Just a thought. Okay, so a couple things I want to remind you of. Every single test, AP Biology is super, super, super insistent that every test has on it graphing, data analysis, experimental design. So just make sure that you're prepared for questions about independent variable, dependent variable, control. Sometimes they call it control group, and sometimes they call it control. Oh, I suddenly forgot the other name. But if you see the word control, they're talking about the control group. Remember, the control group is the normal or the one that isn't being experimented on. So that's going to be your control group. Okay, and then data analysis. You have, anytime from me, I will give you any equations that are needed, but you will be given an equation sheet for your test. So for example, you're going to do surface area to volume equations on this test today and our test tomorrow. And so I will give you the equations for that. So you'll have what you need to know for that. Okay, next, let's go ahead and talk about what they have, and I'll try to demystify some of the complicated parts of these objectives. Okay, first of all, it says the cell structure, subcellular components, describe the structure and or function of the subcellular components and organelles. The first one they mention are ribosomes. Remember that ribosomes are different than most of the other organelles because they are not they aren't an organelle that has space inside. All they are is RNA that's mooshed up into a ball, basically. So ribosomes are where protein synthesis happens. Synthesis means to build or to make. So if you have protein synthesis, it means proteins are being made there. And sometimes ribosomes are floating around in the cytoplasm, and sometimes they're embedded in the rough endoplasmic reticulum. I'm trying to think if there's anything else. Oh, and ribosomes are found in prokaryotes and eukaryotes. So it's an indicator, a possible indicator that eukaryotic cells came from prokaryotic cells because we share that in common. All right, the endoplasmic reticulum, like I said, we have rough and smooth ER. Rough ER is sort of a place for the ribosomes to hang out. It compartmentalizes the cell, and then the proteins that are being made on the rough ER in the ribosomes, not in the ribosomes, with the ribosomes, is transported to other areas of the cell. The smooth ER detoxifies, which means, for example, it helps with alcohol. So breaking apart alcohol, detoxifying alcohol, other poisons that get into the cell. And then it helps with lipid synthesis, which means the building of lipids. The Golgi, now in the italics, it has a little section that just tells you what you don't need to know. The Golgi is that structure that looks like flattened pancakes, and I say it's like pancakes with blobs of syrup coming off. The blobs of syrup are the vesicles that are coming out of or going into the Golgi. And it says the role of the Golgi in the synthesis of specific phospholipids and the packaging of specific enzymes for lysosomes, peroxisomes, and secretory vesicles is not needed for the class or exam. So you don't need to know real specific things, but you need to know generally what is happening in the Golgi. So one of the important things that it's doing is making sure that our proteins are folded correctly, and it modifies the protein by adding little chemical markers to different ends of the protein. And then it packages them into the vesicles, and then the vesicles are, this is an important word, they're trafficked. It sounds fancy, but all that means is that they're moved around. So they can either be pushed out of the cell in a process that's called exocytosis, or those cool walking guys, the motor proteins, they can carry the vesicles to other parts within the cell. But the vesicles themselves are the things that are made by the Golgi, part of the Golgi, and that are used to transport proteins and other products around the cell or out of the cell, to secrete out of the cell. All right, lysosomes are sometimes compared to the stomach of the cell, but never write that on an AP level exam. It's not a precise use of your vocabulary. So they contain enzymes, and specifically they can contain, you can figure this word out, they contain hydrolytic. So if you remember the word hydrolysis, hydro refers to water. To lyse something means to break it. So they include hydrolytic enzymes, which just means they're adding water and they are breaking apart macromolecules. So that's an example of hydrolytic enzymes. So they're good for digestion. They are also, it'll come up again in a minute, but they also don't just digest food that's coming in, but they might digest old organelles. So something that's malfunctioning in the cell, they might digest that. They even release, they break open sometimes, and they digest the entire cell. And that's called apoptosis. And that, again, comes up in a minute on these notes. So a vacuole, sometimes students mix up vacuole and vesicle. Try not to. The vesicle is the little container that comes from the Golgi, and then it's either pushed out of the cell or transported to another part of the cell. Vacuoles are actual storage containers. Like, that's what they do, is they store things. The most well-known vacuole is the central vacuole in plant cells, and it stores water and other ions in it. And the central vacuole in a plant is super, super important. And it'll mention in these learning targets again in just a minute, because it contains so much water that it increases the water pressure inside of a plant cell. Because plant cells don't have a skeleton, they have trouble standing upright without good water pressure. And that water pressure in their cell walls takes place in the central vacuole. All right. Trying to think if there's anything else we need. Okay, so the mitochondria, the mitochondria and the chloroplast are probably the most complex of the organelles, and we're going to spend an entire unit basically focusing on chloroplasts and mitochondria. So we're just getting introduced to them right now, their anatomy, sort of what they look like, and basically what they do. So the two things that are super important about mitochondria and chloroplasts is that they are double membrane. That is an implication that they were prokaryotes at one time. So they're prokaryotes that enter into a larger cell, and the larger cell engulfed them and surrounded them in a food vacuole or food vesicle. And that's why it has a double membrane. So it's got its own membrane and then the membrane that formed because it was engulfed by that larger cell. So they have a double membrane. The mitochondria are important because they synthesize ATP. They make ATP from glucose and oxygen. Now remember, eukaryotic cells that are animal-like have only mitochondria. The plant-like eukaryotic cells have mitochondria and chloroplasts, both. So that's where cell respiration happens. That's where you take glucose and oxygen, you combine them to make ATP. Break them down. Sorry, you don't combine them. Break them down to make them. Other things, the folding of the inner membrane increases the surface area. Oh, super important, because we're going to have all this conversation about surface area to volume ratio. So if you look at both, actually, the chloroplast and the mitochondria, but we're talking about the mitochondria right now. I'll even see if I can make this. Oh, I was hoping I could go in. There we go. Look at all the folds that take place inside of the mitochondria. That is increasing the surface area of the inside of the mitochondria. Those folds are called cristae. And that increases surface area where these reactions are going to take place, which we will learn much more about making the ATP in the mitochondria. And then electron transport and ATP synthesis occur. You're going to learn what what electron transport is a little bit later, not yet, on the inner mitochondrial membrane called the cristae. All right. Chloroplasts, again, endosymbiotic theory says that they were prokaryotes at one time that were absorbed in, and they have that second layer around it because that's the membrane of the larger cell that absorbed them. They are found in photosynthetic algae and plants. They have the double membrane. Within the chloroplast are the thylakoids and the stroma. So the thylakoids, a whole stack of thylakoids is called a granum. Just an individual thylakoid, an individual little, it looks like a coin or something, that's called a thylakoid, so just one of them. And then the stroma is the fluid that's inside, that's floating all around on the inside of the double membrane. And that's where these photosynthesis reactions are going to take place. Membranes contain chlorophyll pigments, which look green, and we'll learn much more about that in an upcoming unit. And electron transport proteins that comprise the photosystems. You don't need to know that yet. So that's coming up. The light dependent, that's coming up. The stroma is the fluid. You should know that. The carbon fixation, you don't need to know. And Krebs cycle, not yet. Those are all in unit 3. So you don't have to worry about knowing those particular topics yet. All right, continuing with cell structure and function, it says explain how subcellular components and organelles contribute to the function of the cell. Organelles and subcellular structures and the interactions among them support cellular function. So these are a few things that I maybe mentioned, but they hadn't already mentioned. So endoplasmic reticulum offers mechanical support. It carries out protein synthesis if it's rough ER on the membrane-bound ribosomes and plays a role in intracellular transport. So the rough ER helps with transporting some of these proteins within the cell. So intra means within. Inter, like the internet, means out. So connecting to the outside world or connecting outside of the cell. So this one's internal. The mitochondrial double membrane provides compartments for different metabolic reactions. The lysosomes contain the hydrolytic enzymes, we already said that, which are important for intracellular digestion, the recycling of a cell's organic materials, and recycling is super important. The lysosomes are able to recycle things that come in, so things just don't get wasted in a cell. It's brilliant. And then the programmed cell death. Remember in our presentation, I don't think apoptosis is mentioned in your textbook. I think it's mentioned later, but not in this unit, but it's definitely relevant for this unit. Remember in our notes, we saw that an embryo has sort of webbed hands initially, and then apoptosis causes the cells in between the fingers to disintegrate. And that's because the lysosomes are breaking open and they're basically digesting that entire cell. That's called apoptosis or programmed cell death. And then vacuoles have many roles, including the storage and release of macromolecules, cellular wastes, and in plants, it retains water for the turgor pressure. All right, here's our section on cell size. Make sure that you understand that the surface area to volume ratio is what determines how well a cell can exchange materials with the environment. So either bringing materials in or getting rid of wastes, for example. So for example, getting rid of carbon dioxide. Your mitochondria are constantly making carbon dioxide. Here's the problem. Carbon dioxide, when it mixes with water, makes an acid. So if you have a really big cell and very little surface area, and you have mitochondria making carbon dioxide, you're going to cause the internal environment of the cell to become acidic. That's a problem. So you need a small cell with a small volume that can quickly get rid of that carbon dioxide so that the cell itself, the pH does not begin to diminish. That's just one example. So looking at this bullet point, surface area to volume ratios affect the ability to obtain necessary resources, to eliminate waste products, acquire or dissipate thermal energy. So a small cell cools down faster and heats up faster, and otherwise exchange chemicals and energy with the environment. The surface, and these are the equations, and you'll be given these equations on my test or on a test in the future. Okay, so the surface area of the plasma membrane must be large enough to adequately exchange materials. These limitations can restrict cell size and shape. Smaller cells typically have a higher surface area to volume ratio. So, in general, we want small cells because we can get rid of waste and bring in lots of nutrients, and it very quickly gets to the middle of the cell, which is what's covered in the cell size lab. How long it takes to get into a large cell, it takes such a long time for the nutrients to get in or waste products to get out if it's a large cell. All right, so small cells typically have a higher surface area to volume ratio and more efficient exchange with materials in the environment. As cells increase in volume, the relative surface area decreases. So the surface area to volume ratio becomes different, it changes, and the demand for internal resources increases. So the bigger cell we have, if we have a really big cell, it's going to have lots of mitochondria in it. It's going to need tons of oxygen, and it's going to need to get rid of tons of carbon dioxide. If we have a small cell, the need for oxygen is less, and the need to get rid of the carbon dioxide is less. And then as organisms increase in size, their surface to area volume ratio decreases, affecting properties like rate of heat exchange with the environment. Explain how specialized structures and strategies are used for the efficient exchange of molecules to the environment. Organisms have evolved highly efficient strategies to obtain nutrients and eliminate wastes. Kind of what we've been talking about. Cells and organisms use specialized exchange surfaces to obtain and release molecules from or into the surrounding environment. I added the examples because I felt like that was a little bit like you're like, what? So here's an example of a specialized structure. The cristae, the folds in a mitochondria, increase the surface area on which reactions can be occurring. So that's one example. And then the many multicellular organisms have developed organs or organ systems like lungs or intestines, for example, to increase surface area contact with the environment. So lungs increase the surface area for exchanging gases. Intestines increase the surface area for the absorption of nutrients. All right, we're getting close to the end. Compartmentalization, ooh, big word, also known as, well, it's part of the endomembrane system. Describe the membrane-bound structures of the eukaryotic cell. Membranes and membrane-bound organelles in eukaryotic cells compartmentalize intracellular metabolic processes and specific enzymatic reactions. So basically, if it says explain how it's important, the internal membranes facilitate cell processes by minimizing competing interactions. So you can have, for example, lysosomes are full of these digestive enzymes. We want those digestive enzymes just here. We don't want digestive enzymes floating all around because that's going to digest the entire internal anatomy of the cell. So we're compartmentalizing so that specific reactions happen in specific places. So, and then we can also increase the surface area where those reactions can occur. The origins of compartmentalization. Hey, let me just say one thing about the endomembrane system. Remember that that's the whole path of how proteins move from, they don't move, but they're started sort of the message from the nucleus all the way out to the cell membrane. Let me walk you through it really quickly. So in the nucleus, the DNA has the instructions for building a protein. So it sends out an RNA out of the nucleus, and that RNA goes to a ribosome, either a bound ribosome on the endoplasmic reticulum or a ribosome that's floating around in the cell. That ribosome builds the first protein. So it's getting built. It reads the RNA and it builds the protein. The protein then, if it's in the endoplasmic reticulum, it might get bundled into a small vesicle and join with the Golgi apparatus. Or if it's out in the cell itself, it still might get bundled in a vesicle, or it might get transported somewhere else. If it joins the Golgi, in the Golgi, it then gets modified. It gets folded correctly. It gets little chemical markers added to it, packaged again in a vesicle. And then that new vesicle either can be pushed out of the cell so the cell can secrete, that's the word, and that's also called exocytosis. We saw that in the videos when the cell pushes the contents of a vesicle out of the cell. Or if it doesn't get secreted out of the cell, it could be carried, that vesicle could be carried by one of those motor proteins somewhere else in the cell. So that's really the endomembrane system. All right, and then finally, the origins of compartmentalization or endosymbiosis describe similarities and or differences in compartmentalization between prokaryotic and eukaryote. Prokaryotes don't compartmentalize for the most part. Eukaryotes do. They have these organelles. Membrane-bound organelles evolved from once free-living prokaryotic cells via endosymbiosis. So that's the idea that an organelle was engulfed by, that a prokaryote, a small cell, was engulfed by a large cell. You should know the evidence for endosymbiosis. So look that up. Evidence might include things like the fact that it has a double membrane, the fact that it has DNA, its size is comparable to prokaryotic cells. There's several things that you should look up and make sure that you know some evidence why we think that some of these organelles or all of these organelles are endosymbiotic. Some of these organelles are endosymbiotic, not all. And ribosomes also are common between prokaryotes and eukaryotes, so that's another idea. So prokaryotes generally lack internal membrane-bound organelles, but they have internal regions. So for example, within their cell membrane itself, they have specialized areas that have special structures and functions. So they're able to do a lot of the things that the big cells are able to do, but the big cells do them in little specific areas in organelles, whereas the small prokaryotic cells do it in a specific area of the cell membrane. All right, and then describe the relationship between the functions of endosymbiotic organelles and their free-living ancestral counterparts. Membrane-bound organelles evolved from previously free-living prokaryotic cells via endosymbiosis. The symbiont provides a metabolic service to the cell. So, for example, chloroplasts provide glucose and oxygen to the cell, while the cell provides nutrients and protection to the symbiont. So the chloroplast is protected inside the cell, and so it's happy to live there. And now it's a permanent structure. It would never live outside of it. Okay, I think that's it. I hope that wasn't too long. I don't have a sense of how long that was, but let me know if you have questions, guys. Have a great day.